My name is Catherine Chen, and I am the Community Science and Outreach Manager at New York City Audubon. Before we get started, I'd like to just address some quick logistics. First, like all of our lectures, this one will be recorded and later posted on our website for anyone to view. Closed captioning is enabled for the Zoom and in the recording, and we'll be sharing the recording with everyone who registered later this week. And second, during tonight's lecture, please type any questions you have in the Q&A. You can open the Zoom feature by clicking on the Q&A icon at the bottom of your screen. We'll take as many questions as we can after the presentation. All right, so for those of you who are not familiar with New York City Audubon and our work, we are a grassroots conservation nonprofit that works to protect wild birds and their habitat in New York City. Through research, science, advocacy, education, and outreach, we work to make the city a more bird-friendly place. One of the ways we do this is by studying bird collisions with windows in the city. And as you'll be hearing about in a little bit from our guest speaker, birds see differently from humans. So understanding how and what they see is an important part of understanding and preventing collisions. Our Project Safe Flight program enlists volunteers to monitor select buildings in the city, and the data that these volunteers collect is critical to making the city a safer place for birds, including the stars of tonight's lecture, hummingbirds. If any of you here live in New York City, and I know I saw some that were from the city, Manhattan, Bronx, Brooklyn, Queens, and Staten Island, we would love for you to join us to help monitor for collisions. Rosalind Rivas, our public programs manager, is dropping a link in the chat where you can find more information on how to get involved in Project Safe Flight or any of our other volunteer opportunities. One of the other aspects of our work is our education programs, which includes this winter lecture series. Tonight's lecture is the last of this series, so if you missed any of the previous 4 lectures, no worries, they were all recorded and are available on our website. Rosalind, again, is going to drop the link in the chat. A huge thank you to Claude and Lucienne Bloch, whose continued support has made tonight's lecture possible. Now, I'm sure everyone is very excited to hear about hummingbirds and their plumage, so without further ado, it is my pleasure to introduce Gabriela Venable. Gabriela is a PhD student at Duke University in Evolutionary Anthropology department, where she studies cognitive evolution. Previously, she had studied ornithology at Yale, conducting research in Mexico on hummingbirds and in New Zealand on parent intelligence with the Kia. She was also a tour guide for 3 years at the Yale Peabody Museum's bird collection and loves a good weird bird fact. Tonight, while not necessarily weird, she'll certainly be sharing many extremely cool hummingbird facts with us, which I can't wait to hear. So with that, please take it away, Gabriela.
Awesome. Thank you so much for that introduction. And thank you so much to all of you for coming out to listen to me talk. I'm always really excited to talk about hummingbirds or just birds in general. Honestly, birds were probably one of my first true loves. I did research on them for about 8 years, hummingbirds for 6 of those years. Currently, I'm not working with birds. I'm taking a brief hiatus. I study how cognition or intelligence evolves, and I'm currently doing that with lemurs. But I'll take any chance I can get to talk more about birds. So again, thank you so much for coming, and thank you for inviting me to speak here today. All right, so why am I so excited about hummingbirds? And why should you be excited about hummingbirds? Why do we even study these birds? Well, hummingbirds are just absolutely incredible. They live life at the extreme. And partially, I mean, just think about it. First of all, these birds are at the extreme of the size spectrum for birds, right? We've got the smallest bird in the world, the Cuban bee hummingbird, coming in at about, I believe, 0.02 grams, or a quarter of the weight of a dime. And despite these small sizes, these birds are able to complete some incredibly long migrations and other tremendous feats. I should mention, actually, the biggest hummingbird in the world, the giant hummingbird, is only as large as a sparrow. So these really, truly are very small birds. But going back to their migration, you probably already know that these birds have amazing migrations, especially those of you who are in New York City. If you've read up a little bit on the ruby-throated hummingbird, one of the most common hummingbirds that you'll see in New York City. And if you've learned about these birds, you probably already know they will fly across the Gulf of Mexico. So this, let's see if you guys can see my mouse. This area on the map right here, they'll fly across the Gulf of Mexico during their migration from the tip of Mexico up into eastern US. They'll go as far up as Maine. So really just an incredible distance for such a small bird, but that's not the longest migration for any hummingbird. There's another bird that sometimes I believe you guys can see as well in New York City. It's the rufous hummingbird. They've got an orange throat. Typically, they won't be as common over there. They're kind of a migrant or a vagrant. We've started seeing them more on the East Coast due to more clear cutting. But usually what they do is they migrate along the West Coast, and they'll go all the way from the Isthmus of Mexico, this purple region on the map, all the way sometimes into Alaska. And that's just an incredible distance for any bird, let alone a hummingbird. So people actually didn't used to believe that hummingbirds could fly that far. Previously, people thought that they rode on the back of geese. So that brings up two different reasons we might want to study these birds. They have an incredibly small body and are able to, despite that, go very long distances. They're also really interesting because they live their lives in the fast lane. Hummingbirds are typically cruising around about 20 to 30 miles per hour. They'll dive at about 60, maybe 50 to 60 miles per hour. Their heartbeat is going about 1,000 beats per minute. To put that into perspective, our resting heart beat is about 90 beats per minute. They've also got their wings going about 80 beats. Ooh, is it per second? I'm gonna say per minute to be safe there. But yeah, they're living life in the fast lane. And in order to maintain such a fast lifestyle, they have to have a very fast metabolism. And that's one of the reasons people are very interested in them. And it's something that causes a behavior that we notice a lot at feeders. You'll notice that whenever a hummingbird is at a feeder or a floral resource of any kind, they're really defensive of these resources. They're always constantly fighting. And that is because they have a really fast metabolism. In fact, I believe it's if a hummingbird doesn't have a fat storage already built up, I think they're within 30 minutes of starvation. So in other words, these social interactions we see, this fighting is actually a matter of life or death. These birds are just trying not to starve. So what does that mean? We're very interested in studying them for their metabolism, for their social interactions. An important trivia fact, if you go to bird trivia, hummingbirds are the only birds that can fly backwards. So they're also of interest for their locomotion as well. All right. Another reason why we typically are interested in studying hummingbirds is because they have essentially figured out how to live in a very diverse, different set of habitats. We've got hummingbirds that live in the desert, hummingbirds that live in the rainforest, hummingbirds at the top of mountains, at low altitudes. These birds that live at the top of mountains are living in very low oxygen environments. So how have they figured out how to live there? Well, these are all, this is all very interesting. You can think about it. Hummingbirds have learned how to live in all these different environments and have adapted specialized traits in order to live in these different environments. Additionally, we study them because of their evolution. So hummingbirds are a very diverse group of birds. In the US, we have about 20 species of hummingbird, give or take, maybe a few less. But there are many, many more species. There are about 330 species of hummingbird in the world, and they show a lot of diversification. So They've gone to many different habitats, as I've mentioned before. Some groups within hummingbirds, some species have shown, some different groups have shown rapid speciation. Some have shown slow speciation. They're just very interesting to study. How did they get to be here? Hummingbird fossils have been found in Europe, and yet now we only find them in the New World. So we really are truly lucky to have hummingbirds around here. Anyone in the old world, so in Europe, Africa, Asia, can't say the same. They don't get to have hummingbirds. All right. But the reason why I'm here to talk to you all today is maybe one of the coolest things for hummingbirds, for me at least, that's their coloration. So hummingbirds are incredibly colorful, and this is reflected in their names. We've got mountain gems, sapphires, emeralds, ruby-throated. They're just so incredibly colorful, and that was one of the reasons I wanted to study them. I wanted to look at how colorful hummingbirds are, and whether or not that could help us better understand how colorful birds are in general. All right. Now that I've hopefully convinced you that you should love hummingbirds if you didn't already, I want to talk a little bit about plumage coloration before I get into the work I did looking at hummingbird color diversity. So plumage coloration is very important because it defines where a bird stands out or blends in. So coloration is important for camouflage, but it's also very important for communication. And there are a variety of different types of communication that hummingbirds use their color for. So they'll use it for territoriality and defense. You've probably seen this at a feeder. Maybe there's a lone male guarding a feeder. Another hummingbird comes in. That male that's guarding the feeder will puff out its feathers, fly towards that intruder as if to say, hey, I am here, get out of here. This is my floral resource or my feeder. And of course, color is also very important for identifying the different sexes and for sexual display. And I include identification of sexes because just as we as birders are looking at hummingbirds and trying to distinguish them according to their colors, so are the birds themselves. And color is very important for sexual display. Sexual selection is very important in hummingbirds. We get a lot of ornamentation, whether it's colors. We also get those beautiful long tail feathers that I mentioned before. All of this to woo a female. And hopefully this video will work. Hummingbirds have evolved very elaborate displays. So not only those long tail feathers, but also they have evolved specific feathers that when they dive make whistling noises. And also they just do very elaborate displays where they puff out these brilliant patches of colorful feathers, like we will see hopefully right now. So here we have a female and there's the male. So this is a bird from back home, actually, for me. I'm from Tucson, Arizona, and we just grew up with these in our backyard. This is the Costas hummingbird, but just look how it really is using those colorful patches of feathers to try to woo a female. It's just absolutely beautiful. All right. And keep on going. All right. And something I should mention, though, is, and it was brought up before in the introduction, birds don't see color the same way that we do. So we see 3 different types of light or 3 different types of color. We see red, green, and blue light. And that's because we have 3 different specialized types of cone cells in our eyes. 1 cone cell type for red, 1 type for green, 1 type for blue. However, birds have a 4th cone cell type, and that allows them to see ultraviolet light. It is because of this that we say that birds perceive the world very differently from us. There are other reasons too, but it's important to realize this when talking about color because they're perceiving color completely differently from how we do. So you can imagine if we could only see the colors green and blue, how amazing it would be to suddenly be able to see red and combinations of red, blue. You suddenly have purple, a completely novel color that if you only could see red and green, you would not be able to imagine. And so that's kind of the case when we're trying to imagine what colors look like for birds that have an ultraviolet component in them. So here is kind of an image getting at that. So here on the left, we have a cockatiel and a chicken egg, as we would see it. So with our red, green, and blue receptors. And then here's an image in the middle that shows what part of the cockatiel and the chicken egg reflect ultraviolet lights. And then on the right, we have a simulation of what it might be like to see colors as birds do, as a combination of red, green, blue, and ultraviolet lights. Another important thing to know about color is in the natural world, there are two broad classes of color, and they're defined by how they're produced. So we've got pigment colors and structural colors. Pigment colors are created by molecules within the feather, within a structure. And the molecules themselves are the color that is produced, that we see on that surface. So for instance, imagine the dyes in your clothing. If you have a green dye and you use it to make clothing, you're going to have green clothes, right? That's kind of how pigments work. So pigments produce the browns and blacks, the reds, oranges, and yellows in birds. They do produce a few other colors as well, but pigments really are limited in terms of the flexibility of color production and the colors that they can make. So some examples of pigments that we have are melanins, right? Melanins are what give us our skin tone, our hair color. Yeah. On the other hand, we have structural colors. Structural colors are colors that are created by the way that light interacts with microstructures within the surface of whatever, in this case, a feather, right? So in this case with structural color, the surface of the feather doesn't have pigments the same color that's being produced. The surface isn't the same color as the color that's being produced. So that's a little hard maybe to understand. So I have a few examples. Imagine a prism, right? A prism is clear, but the way that light interacts with it causes it to produce almost like a rainbow, right? A spectrum. And so you see every color from red all the way down to violet. You can also imagine this with an oil slick. So oil is typically brown or black, but once you put on water, it has an iridescent sheen. And that's because of how light is interacting with the oil and water layers. And that's how structural color works. And that's also the case here with this blue jay. So a blue jay feather, or at least the blue feathers, the pigments in that feather are actually brown or black, but it's the way that cells in the feather are arranged and how light interacts with those cells that produces a blue color. All right, so hummingbirds have a very particular type of structural color, and it's called barbial structural color. Barbial just refers to what part of the feather is producing the color. So imagine a feather is like a tree. You've got the trunk in the middle, or like the main vein of the feather. Then you've got branches that come off. Those branches are called barbs. And then off of those barbs, there are even smaller branches. And those smaller branches are called barbules. So that's where barbial structural color is produced, on the barbule. And what's important to note about, well, I guess structural color more generally, first of all, structural color more generally is able to produce a wider range of colors. It's a much more flexible mechanism for producing color. Barbial structural color is even more flexible than other types of barbial structural color in that it allows for the easy production of color combinations. So like red and blue being purple, or ultraviolet and green, an ultraviolet green color. And it also helps produce very saturated colors or very intense colors. And that was one of the reasons why I wanted to study hummingbirds. Hummingbirds, all these brilliant colors that they have are being caused by them having barbial structural color, or that's the mechanism behind it. And previous studies looking at how colorful birds are didn't sample as many barbial structural colors. So I thought that if I looked at hummingbirds and had a better idea of how colorful they are, measured their total color diversity, we could have a better understanding of how colorful birds in general are. And that was my senior thesis at Yale. And so now that I've kind of given a little bit of an introduction, I want to talk a little bit more about what exactly I did and what I found. But first, let me explain what exactly I wanted to look at. So I was interested in measuring the total diversity of hummingbird plumage coloration. Then I wanted to compare this diversity to the previously known color diversity for all birds. Then after that, I wanted to calculate the new total diversity of color for all birds by combining the data from a previous study on avian coloration with the data I collected from hummingbirds. All right, so how did I go about doing that? How do you even measure a color? What does color diversity mean? How do you measure that? All right. So I have a broad statement here. I'm going to explain what it means, but first I'm going to say it. So I measured spectra from 114 different species of hummingbird and collected over 5,000 spectra. What does this even mean? So I measured colors from 114 different species and collected data on over 5,000 spectra, or sorry, 5,000 colors. How you go about that, though. So typically, when we measure color, we use a device called a spectrometer. A spectrometer tells us which wavelengths of light are, and wavelength defines what kind of hue, what color we see, what color we see. So like longer wavelengths are red, shorter wavelengths are blue. So spectrometer tells us what wavelengths are reflected off of a surface and at what intensity. And the intensity tells us how saturated a color is. All right, so I would take a spectrometer like this box here on the upper left, and I would point a little light probe. It's this blue wire coming out of the spectrometer, point it at a plumage patch. It will direct light towards that plumage patch and then collect wavelength data, or collect the wavelengths that are reflected back, and again, tell me what wavelengths are being reflected back and at what intensity. And this is a graphical representation of what that data would look like. And so from this graph, what I can see is what color is being produced and about how saturated it is. So for this color, I can see that there's a peak at about, we'll say maybe 670, 680, so red, and also a peak around 450, which is blue. So what's being produced here is a purple color. And the same thing if we look at this green one. So it's about 530 and then about maybe 390. That's an ultraviolet green color. All right, and so that's how you measure a color. So once I had all of this data on color, I could then quantify color diversity. So to do this, though, I wanted to take into account the fact that birds see color differently from how we do. So you can imagine that if I wanted to model all the colors we can see, I mentioned before, we can see 3 different types of light, right? Red, green, and blue, because we have 3 different types of cone cells in our eye. Well, imagine we have a triangle, right? And at each vertex, we have the maximum saturated color that we can possibly see for red, green, and blue. Well, the area of that triangle could be a representation of all the different colors we can see. Now, if we extend that to birds, we can add an extra vertex for ultraviolet light. Now we don't have a triangle, we have a pyramid instead, or what we call a tetrahedron. And that represents all the colors that birds can see. So then I take my data on color and I plot it as a single point within that pyramid volume. And the distance away from the center defines the saturation of the color, or how intense that color is. And the angle in respect to the different vertices tells me what color I'm seeing. So let me add some color labels onto this. So I've added these color labels, these little circles. We've got ultraviolet, red, green, blue. If we look at this little gray dot here, it's, you know, partially between blue and green. It's pretty far from the center. So that would be a fairly saturated turquoise color, or blue plus green. And so then I plotted all of the different color measurements I took within that 3D space. And I measured the minimum volume covered by all of those color points. And that is my measure of color diversity. So again, plot out all those color points, and the volume covered by them is a measure of color diversity. Okay, so we understand now how you measure a color and how we can calculate or estimate color diversity. Let's see if we can get out of here. There we are. All right. A side note, where do you even find 115 species of hummingbird? Well, I wasn't working with wild birds. I wasn't working with living birds. This was purely done with museum specimens. And I was incredibly fortunate because I had a wonderful resource at my fingertips, the Yale Peabody Museum of Natural History, a fantastic museum, really. If you can visit it, you really should. And they have a great research collection. So I was able to go in and find a bunch of different species to study, but they didn't have everything. So I actually ended up going towards your neck of the woods. I was at the American Museum of Natural History in New York City. I'm sure you're much more familiar with that area or that location than I am. I mostly spent all my time, if I can get back there. I spent most of my time really in a small dark room measuring bird skins. So I'm sure you know the museum better than I do. But yeah, this is where I like to say that natural history museums, like the ones that you go to, often have a public collection or a public display, which is what you guys normally interact with. But there are also often research collections in the background, which are incredible resources for students, artists, or researchers like me. Without these types of collections, there's so much different work that we couldn't do, including a lot of really important conservation work. And I also want to give a really quick shout out to Yale Peabody Museum. I mentioned this natural history museum is great. You guys are pretty close by. I think it's like an 1.5-hour train ride. If you happen to be in New Haven and you're looking for something to do, consider stopping by. They have a really great behind the scenes tour. I'm not sure if they have any upcoming or when the next one would be, but it's a great experience. You learn about so many different species. You get to see specimens of species you probably wouldn't see otherwise. And you learn so much about the different species in the collection, about the research that's being done with these species, or these specimens, I should say, as well as the conservation work that museums like this have done. All right, so what did I find? Okay. So what might be a little bit surprising to y'all is that despite how colorful hummingbirds are, they only produce about 1/3 of the colors they can see. Why? All right, so there are two potential reasons why this might be. So first, it could be that certain colors are selected against because they have low signaling value, or maybe there are certain colors that are just hard to produce. So for that first explanation, certain colors are selected against because they have low signaling value. What that means is there might be some colors that aren't that good for communication. So you can imagine that if you live in a fully green environment, it might not be very helpful to evolve green plumage coloration for communication because no one's going to see green on a green background. So in other words, green would have a low signaling value. It's also possible, though, and I will argue the second, that certain colors are mechanistically hard to produce. And I'll explain why I think this is in the following results. Right. So the second result I found is that hummingbird color diversity that I calculated was greater than the previously known color diversity for all birds. All right. So the sample that I looked at for hummingbirds was more colorful than the sample for all birds that was previously done. So if you look at these figures, let me give a little bit of reference to what you're looking at. So in black, we have the previous study that was done looking at the previously known diversity of color for all birds. And then in blue, we have my hummingbird color diversity that I measured. All right, now let's go over to these triangle figures over here. So this is a view of that pyramid space that I showed before, but we're just looking at one plane. We're just looking at one side where you can see really well where things are oriented in terms of the red, green, and blue vertices. All right, and so what you'll start to notice is that hummingbirds are more colorful, or they're accomplishing a greater volume within that color space because they're going into regions of that color space that weren't as common in other birds, that were kind of rare. So those colors include very saturated blues, very saturated greens, and some color combinations like saturated purples, which is a red and a blue, or saturated turquoises, which are green and blue. All right. They also were able to create some other unique colors. Let's see if I can get that, such as UV green. And these figures over here, these map-like figures, are maps of hue. So we're above the green triangle here, which means we're in ultraviolet space and in the green area. And what you see with the blue, the figures here in blue, is there are a few points here in a space that isn't really occupied by all birds in the previous study. All right. And I want to focus in on these ultraviolet green colors. Okay. So over here on the left, we have that same hue map. And I have it mapped out in a tetrahedron model, so an avian color space. And if you look at the center, you can see where it's plotted. It's A1 and A2 right here. But if you look at the right, you can see that that same area is empty for the previous study looking at how colorful all birds are. And I also, the spectra that you've been looking at this whole time, or this graph you've been looking at of color, of different wavelengths, this whole time has been actually this ultraviolet green color that's here. All right. And the reason why we believe, and my collaborators would agree, we believe why we're seeing colors like ultraviolet green that we're not seeing in that previous sample, as I mentioned before, is because they're hard to produce. All right. So if you look at this figure here, I want you to first ignore these lines, right? Ignore the purple and green lines. Focus first in on the boxes. We've got 4 different boxes. Each one represents the range of wavelengths that each cone type is sensitive to. So red, green, blue, purple. So for that ultraviolet green color, now looking at that line, what you're noticing is how well it fits that green and that UV bin. So to produce a really saturated, pure green UV color, you have to have very tall peaks because the tallness of the peak is what defines how saturated that color is. But they also have to be really narrow, because if they're not narrow, they'll start to stimulate the neighboring cones, right? So you have to have a very narrow peak in the green that doesn't stimulate the red or the blue, and you have to have a very narrow ultraviolet color that doesn't stimulate the blue. That's also true for saturated purple. You have to stimulate the red cone and the blue cone, which out-stimulating the neighboring cones. And it's also true for blue and green. Does that sound familiar at all? Maybe, because I just brought those up as being colors that were rarer in the sample for all birds, but that we see pretty often in the hummingbird sample. So in other words, it might be that hummingbirds have a method for creating really saturated color combinations as well as single colors that are difficult to produce just because those peaks have to be really narrow and really tall. All right. So altogether, hummingbirds increase the total avian color volume by 56%, which is pretty cool. And these figures over here on the right kind of show exactly what I showed before, that hummingbirds, which are in gray, are going into regions of color space that we didn't see with the previous bird sample, which is in black. We also looked at which hummingbirds were the most colorful and some of the species that showed these unusual colors that were going into regions of color space that we hadn't seen before for the previous study on all birds. The most colorful hummingbird species, for anyone who's really into hummingbirds, was actually this individual here, labeled B. That's the violet purple coronet. And it was able to accomplish a really large color volume or color diversity because of that purple patch on its belly. That what we see as purple is actually just a little bit of violet that's coming off of a very saturated ultraviolet peak. So this is a pure ultraviolet color. And this was the only species I found had a pure ultraviolet color, which is really cool. Hummingbirds in general didn't use all that much ultraviolet compared to other birds. The second most colorful species is this one right here in A. That is the fiery-throated hummingbird. Not too surprising. It's like a rainbow. And then some other notable birds that I should mention, or one more notable bird I'll mention, is this one, G. This is Geoffrey's daggerbill. And we've actually been looking at the spectra for this bird this whole time. This bird produced this saturated purple and this saturated UV green. And it was one of the few birds that produced a saturated UV green. I think there were only 2 species that did. All right. And if you want to know more, there's a figure in the paper that I wrote that goes more into that. An important other thing to note is that the most colorful plumage patches on hummingbirds are those that are most prominently used for communication. So that would be the throat and the crown patch. And these are the patches that are immediately most visible when individuals are interacting with one another. So when an individual is facing another individual, they're going to see very clearly very saturated crown and throat patches. And so that shows us the role of selection for communication on hummingbirds. All right. But why are hummingbirds so colorful? So why am I getting these results? All right. So something I mentioned before is that, hummingbird evolution, right? So hummingbirds are a very species-rich group of birds. And when we see speciation or different species are forming, sometimes we'll see selection for differentiation between elements that are very important for mating displays. So for instance, color. And these serve as boundaries or barriers to prevent, that help prevent interspecific breeding or different species from breeding with one another. And so in a very species-rich group, you might imagine that you might see a lot of color if color is very important for getting a mate. And there is a paper that found a similar result. There is a paper that actually found that color evolution rates correlate pretty tightly with speciation rates in hummingbirds. I also brought this up before. Hummingbirds rely heavily on plumage coloration for communication, especially for sexual display. And so this really gets at, and the finding that it was the throat and the crown patch that were the most colorful really gets at the point that there's been a lot of years of very intense social and sexual selection on these birds, which has probably led to there being such a large color diversity in this family. Lastly, getting back to mechanisms and talking about barbial structural color, these birds have a very unique type of cell in their barbules that's both hollow and oval shaped. And it's the shape and hollowness of these cells that allows hummingbirds to create very saturated colors. So like those blues and greens that were rare in the previous sample for all birds, but also lots of color combinations. So UV greens, UV yellows, UV reds, all sorts of things. It's a very flexible way to create new colors. And yeah, so those are the two main reasons why hummingbirds are so colorful. Anywho, I want to quickly acknowledge some people that were really important for this work. I need to thank my former advisor, Richard Prum, who helped get me started on this project. He told me I should probably consider looking at hummingbirds for their coloration, and I really have to thank him for that. There's Kaya Gam, who helped do all the R programming I needed to be able to analyze my results. Krzysztof Zygowski, head of vertebrate zoology, or one of the heads of vertebrate zoology at the Yale Peabody Museum, who is like a secondary mentor to me and also just a great friend. Bentley Bird at the American Museum of Natural History for helping me find everything I needed at the American Museum of Natural History. And then head David Evans of Berkeley College and David Heiser at the Yale Peabody Museum of Natural History for finding me funding resources. And then more importantly, again, thank you guys for listening to me blabber on and on about hummingbirds. I'm always really excited to talk about these birds. And I really hope that I've given you just at least one more reason to love these birds just a little bit more. And my last thank you, I give you one of my favorite types of hummingbirds. Some hummingbirds have puffy feathers around their legs, little booties. And I love them so much because they don't do this to keep their feet warm. Rather, they have these little boots because females find them attractive. Yeah, that's all I have. Thank you.
Thanks, Gabriela, for that amazing talk. We did have some great questions in the chat and in the Q&A. So our first question is from Kathy, and she asked, do we know why hummingbirds are no longer in Europe, Africa, and Asia?
Ooh, yes, I think we do, but I can't remember. So I believe it had to do something with they came over due to the opening of some sort of environment with novel floral resources, but I can't say exactly what those were. But that's a great question.
Awesome, thanks for that answer. The second question is from Jane, and they ask, can all birds see UV light?
Okay, let me think. I'm taking a minute to think about that. It might be there. I'm trying to think. So I know that all diurnal birds can see ultraviolet light, but I'm not sure about nocturnal birds. But your average bird, yes, will be able to see ultraviolet light. Some can see ultraviolet light that is of shorter wavelength. So I showed, I wonder if I can get there. Let's see. So yes, they can see, there we are. So different types of birds might have ultraviolet cones that can let them see further, like over towards 330 nanometers. But yes, the average bird and birds in general can see ultraviolet light.
Our next question is from Jean, and she actually asked two questions. The first is, did your spectrograph measure only backscattered light, or did you measure it at different scattering angles?
Yes. All right, so let me get at your question. So only backscattered light, or did I measure at different angles? So I think this gets at your question, if I understand it correctly. So I used only one angle for all my measurements, and I used whatever angle gave me the maximum saturation for the coloration I was looking at, because that's a way you can kind of standardize measurements. There is only one angle that will allow you to reach maximum saturation, and that should be about 90 degrees from the color producing structure or layer.
And then their second question was, would a spectrograph measure color from pigment versus spectral differently? And how did you deal with that?
So from color or from structural color differently? Yes, so that's actually a really great question. So a bunch of different things come to mind. So yes, for iridescent colors, let's see. Let me think. So, okay, and the real answer is no. The real answer is no. If you look at a pigment versus a structural color, you're going to get the same kind of output. However, with iridescent colors, like the ones we see in hummingbirds, it can be more difficult to measure color. So yes, but it might be a little bit more difficult. Hopefully that helps.
Let's see, Jane and Maya ask, does aging affect color, and do the colors fade in the specimens?
Yeah, that was exactly why I was wondering whether I should cover in the last question. So structural colors don't fade. Structural colors don't fade because they're not being created by molecules that deter with time. So pigments, yes, pigments will fade with time, but structural color will not for the most part. And also, there are some specimens that were absolutely blasted with terrible light, because we have some specimens that are over 100 years old in these museums. I just didn't use those birds. I used birds that were more recently collected.
All right. Also, related to whether things affect color is, does pollution affect color, and what about flower pollen?
That's a great question. I'm not sure. I would imagine that pollution could affect color in birds, because color can sometimes be a measure of health in birds. For pollen, not really. It would be like a temporary thing, right? So you can imagine there being some pollen being on there, but you could clean the specimen. But great questions.
Great. Next question is, where do hummingbirds nest and lay eggs?
Ooh, that's a great question. Wherever they can find somewhere safe. So it'll depend on like what species you're looking at. And I'm laughing because sometimes what they consider safe isn't safe at all. Pretty much, yeah. I mean, they'll do it, they'll go in like bushes. I've seen them, trees, just in vegetation in general. But keep an eye out. You never know when you'll find them. You'll find this tiny, tiny little nest, and then like some Tic Tac like eggs just hiding inside.
And then last question that we have for now is, when a hummingbird feeder is hung in a new location, how do hummingbirds find it? Do they primarily rely on sight? Do they have a sense of smell and taste?
All right. Yeah, that's a very, very good question. So hummingbirds and birds in general rely mostly on visual signals. So birds don't have much of an olfactory bulb or a part of the brain that's used for olfactory signals or smells. But they have a very large visual part of the brain. So they primarily focus on color. I could imagine maybe there might be some social learning going on. So you could imagine that a hummingbird might see another bird zip over with purpose to a feeder, and they might learn that way. But yeah, they primarily will use visual cues. And that's why we have red feeders. And that's also why I should also mention hummingbirds are just attracted to red. So red flowers will bring hummingbirds. So visual cues.
Next, we have another great question. Does coloration lead to an increased risk of being preyed on?
That's a great question. It can. And that is considered one of the reasons. Okay, so one hypothesis about why males are so colorful can be that it can be risky to be very colorful. You might have trouble avoiding predators. And if you've managed to get to age of reproduction and you have all these incredibly flashy colors, or even flashier colors than others, then you must be just really good at something. You must be a very viable mate that is very healthy, that can confer those benefits to your young.
Then we have another question specifically about you and your research. Will you be building further upon this study, or are there related questions you hope others will explore after reading the study?
Yeah, there definitely is. Our questions are related to this. So I only looked at hummingbirds, and I was able to find that we were able to greatly increase the total diversity of all colors that we know exist in birds. So it'd be great for other people to go out and study other color-rich groups. We've got parrots, we've got sunbirds, we've got tanagers, New World tanagers. Otherwise, some other really cool things to look at are that how are hummingbirds producing some of these really unique colors? So I mentioned before that the velvet purple cornet in bee has a very saturated UV color. It could be really cool to use an electron microscope to dissect a feather and look at the structures that are creating that color, how that's being accomplished. Same with G, the Geoffrey daggerbill. That was the hummingbird that was producing these curves here. These are really unique outputs for color. This is a really unique color because if you look at it, it's like waves, but they're pretty regular. It almost looks like a harmonic pattern. If you know a little bit about, if you still have some physics background. And we haven't found, this was the only species that had anything like this in terms of the color it was producing, that wave-like pattern. And there aren't too many examples of other birds producing colors like this. So there might be something really special going on with this species.
Great. Thank you so much. I see that we do have a couple more questions in the chat, but unfortunately, I think that's all the time we have for today. If you really want to know the answer to your questions, Gabriela or Rosalind will be dropping Gabriela's email in the chat. Please feel free to follow up with Gabriela for any questions that you have that have not been addressed yet. Thank you so much, Gabriela, for this fantastic talk. And thank you, everyone in the audience who's came and attended and asked questions. I know most, not if all of us, will agree with me when I say that I learned so much from this talk, and I learned so much from the questions that were asked after the talk was completed. The recording of this lecture will be posted later this week at the link that Rosalind is dropping again in the chat. This is where you can also view recordings for all of our previous talks in this winter lecture series. If you have been inspired and would like to go out and see the hummingbirds and other birds in our own backyards, we at New York City Audubon offer hundreds of bird outings annually. You can see our calendar with over 200 upcoming spring bird outings, classes, and festivals using the link that Rosalind is posting in the chat. All right. Thank you so much for joining us again. And another big thank you to Gabriela. Have a great night, everyone.
Thank you.